All right, welcome back. So we'll be able to talk about the prophase, and we've talked about the metaphase. We said the phase existing between the end of one mitotic division and the beginning of another, the interphase, where we talked about the DNA replication and duplication as the major activity that occurs there. Then at prophase, we talked about the chromosomes become bivalent, right? That's the major things that occur there. Then the nucleus and nuclear membrane, they disappear. Nuclear membrane breaks down, nucleus reappear, right? And then when that happens, what happens? This nuclear content now floats in the cytoplasm, and then the bivalent chromatin lie along the equatorial plane at metaphase. So now we're going into the next phase, which is anaphase. So now you can see at metaphase, they line along the equatorial plane, right? So at anaphase, there will be a splitting here. At this centromen, there will be a splitting at the centromen. The centromen will split longitudinally, alright, so that these chromatids become separated. They become separated chromatids. They become independent chromosomes at anaphase. So at anaphase, they are separated now and they become independent chromosomes, alright? And then they begin to migrate to the opposite pole along the spindle fibers, alright? So that's what occurs at anaphase. That's what occurs at anaphase. At the centromen, there is splitting longitudinally, separating the two chromatids to become independent chromosomes, and then they begin to migrate to the opposite pole along the spindle fibers, alright? So they get to the opposite pole at telophase, and when they get to the opposite pole, what happens? Remember, the nuclear membrane breaks down and the nucleus disappears at prophase, but at telophase, when these separated chromatids, now chromosomes, get to the opposite pole, what happens? The spindle fibers will break down. You know, the spindle fibers is formed by this central. They break down, and then the nuclear membrane and the nucleus reappears. Once that occurs, you can see that the cells start taking shape. All cell organelles within, they duplicate, alright, so that each cell will have the same cell organelle. That is, they have similar number, similar type of cells on each, similar type of cell organelles on each, alright? So there have to be duplication of the cell organelles, alright? So that's what occurs at telophase. You can see that at the telophase, there's a constriction that will occur here. A constriction comes in at this telophase, and this is where the cell will split. That's where cytokinesis comes in our next discussion. We'll talk about that soon. So at this telophase, what happens? The spindle fiber breaks down, the nuclear membrane and the nucleus reappears, and then these chromatids, now chromosomes, they were once rod-like, right? They were once rod-like. So what happens? They begin to elongate, and once they begin to elongate, they begin to turn thread-like again, alright? So that's what occurs. So now once this telophase is over, this is the end of mitosis. Remember, we said mitosis comprises of prophase, metaphase, anaphase, and telophase. So at the end of mitosis, that's telophase, what happens? The next stage is cytokinesis. And remember, we said cytokinesis deals with the division of the cytoplasm itself, alright? Division, so that it results in two daughter cells as in the case of mitosis, or 4 daughter cells as in the case of meiosis. So what happens here? This contractile ring, there's a contractile ring that will exist at the point of cleavage which is at the constriction. There is this contractile ring. So at this region, this contractile ring is what will cause the cells to cleave. There will be a cleavage at this point of constriction, so that the cells become separated. So you have two identical daughter cells here. Alright, so that's it for this cell division. So far what we're going to talk about is what mitosis is. Alright, then interphase, what's the major thing that occurs there? DNA duplication occurs here, where the DNA within these strands, within these chromosomes duplicates. Here the chromosomes are what? Are not visible, right? They are long and thread-like, are not visible. That's what's happening here at interphase. So at prophase, what happens? This centroid, they separate and they begin to migrate to the opposite poles, and as they migrate, they begin to form microtubules, which will later form the spindle fibers. Then already these chromosomes now become rod-like. Alright, they become more and more coiled, and then they turn rod-like in appearance. And because of this DNA duplication here at interphase, what happens? The chromosomes split into two chromatids held together by the centromere. And then late prophase, what happens? The nuclear membrane breaks down and the nucleus disappears. That's what occurs at prophase. Metaphase. Now remember we said the nuclear membrane what? Disappears. So here the nuclear membrane, there is no nuclear membrane, and the spindle fibers has been formed by the centrum. So the bivalent chromatids come to line along the equatorial plane of the spindle fibers. That's what occurs at metaphase. Now at telophase, there will be a splitting. This centromere that connects the chromatids, it splits longitudinally there, so that the chromatids become separated. They become independent chromosomes. Alright. So now they are independent, now they become separated, and as they become separated, they begin to migrate to the opposite pole at anaphase. Alright. That's what occurs at anaphase. Then at telophase, they've gotten to the opposite pole, and then the chromosomes now, they begin to elongate again, go back to their original shape, and then the nuclear membrane reappears, nucleus reappears, all cell organelles, they duplicate so that this cell has it and this cell has its own. Alright, then a constriction will occur, which will initiate cytokinesis. Alright, so this cleavage is what will initiate cytokinesis, the division of the cytoplasm, so that you have two cells. So at telophase, this is what happens. Alright, then the telophase is the last stage of mitosis. Once telophase is over, the cell now is ready to go into cytokinesis, which is the division of the cytoplasm, so that the cell becomes two. If it's meiosis, first meiotic division will occur and then second will occur. Alright, just like this, but it will result in 4. But now we're talking about mitosis, so it's going to result in 2 identical daughter cells. Alright, so what happens at cytokinesis? There's a contractile ring that will occur, that exists at this point of constriction. So this contractile ring will cause a cleavage here, to cause a cleavage, and then the cell splits and it becomes 2 identical daughter cells with similar content, similar structures, and everything intact. So once that occurs, the cell is ready to grow. It starts growing, getting nutrients, and then it gets to its maximum size, go back to division, just like that. But there are some cells that once they are formed, they go back into division. Alright, those cells are called labile cells, like the cells of your skin. Alright, so those cells, they are labile cells, they go into division, continuous division, they don't need to grow. Then there are some cells that once they go into division, they go to an extent, they keep going. Some of them may not go into division again. Alright, But under stimulation, alright, under stimulation they go into division. Senescent cells, those are cells of your GIT, alright, cells of the liver. For example, if there's a damage to any lobe of the liver, that it regenerates, right? A stimulus causes it to regenerate. The cell division will occur, and that part comes back again. That's cell division. Then there are some cells that don't go. Once they divide once they are formed once, they don't go into cell division again. Those cells are called permanent cells, like the cells, the nerve cells. Those are permanent cells, alright? So that's it for the mitosis. So in the next class, we'll be talking about meiosis. Have any question? Drop it in the comment section below. We'll respond to them, alright? You have topics that we want us to tackle? Drop it in the comment section below. We'll tackle. So it's good to have you guys around. So we'll be talking about meiosis in the next class.